how well you have been doing since you've arrived in India. And it's really a pleasure for me to be chairing this session today. So Dr. Debojyoti Chakraborty did his master's from what was then WBUT here in Kolkata, and then his PhD from the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany. Then he was there in Germany for some time, and then he joined CSIR at the IGIB Delhi as a scientist fellow. And he has been a senior scientist there since October 2016, working on RNA biology. And during his brief tenure here in India as an independent scientist, he has done remarkably well. And his numerous awards in this short period of time are enough to prove that. So he has been awarded the INSA Young Scientist Award, the Young Scientist Award of the CSIR, the Wellcome Trust India Alliance Early Career Fellowship, and so on and so forth. I don't want to spend too much time on that. When you hear him, you will learn for yourself that he is a captivating speaker. And I would just like to remind Debojyoti that you have a heterogeneous audience here. So there are physicists also listening to you. So, and everybody is excited and eagerly awaiting to hear about FELUDA from you, which I'm sure you'll speak about. So with that, it's over to you, Debojyoti. I think from what Jhumu told me, we have 35 minutes for you. And keep some time aside so that those who are listening can also ask some questions. I'm sure you'll be happy to answer them. So welcome, Debojyoti.
Thank you so much, Dr. Bose. I'm audible, right? I see some background noise, probably not from my end. Okay, so it's an absolute pleasure to be speaking here for multiple reasons. I'll tell you one reason at a time.
Just a moment, Debojyoti, if I could interrupt. I think those who are not speaking, please turn off your microphones. That will lessen the background noise. Please carry on.
Okay. Yeah. So, like I said, that it's a absolute pleasure for multiple reasons. One of the reasons is that it's a kind of an homecoming for me. Several of the very distinguished people who are, I'm really thrilled to see some of you actually, because they are mentors. They are, I would consider them mentors. Probably we have not interacted directly, but I was a big fan of Pinak sir's work. And I was an intern in Bose Institute in Pratima Sinha's lab from 2008 2009. So I met him several times on the hallway, and it's absolutely fascinating to see him here in the audience. I see, and also several other people whom I have been in touch with on different occasions. So Bose Institute is a kind of coming. It's a homecoming, because the most of my master's dissertation work was done in Bose Institute Biochemistry Department, first floor. So it's a good fortune that I have been invited. Last time that I had a chance to talk to you, Professor Basu, was of course in 2016. I think I gave a small talk at that point of time for when I was looking for positions here. So this is also the second time that I get a chance. So thank you again to the organizers, Dr. Ghosh, Dr. Dostidar, and I think the expectation is that I talk mostly about FELUDA, so I'll keep the talk on FELUDA mostly. Of course, that is only a very tiny part of the work that we are doing. So, but it's interesting because it's important to understand how CRISPR has really gone beyond just the possibilities of genome editing. Just last, I think day before yesterday or 2 days back, we have been asked also from policies to 10 at the UK strain, and the whole lab is all totally worked out to get this film.
Maybe he has lost internet connection.
I think so. Yes.
Yeah, we'll wait for that.
I think you probably lost me, right?
Yes, briefly, yes. So, are you with us
Hello.
now? Hello.
Can you hear me?
Yes, right now, yes. So, are you with us now?
Yes. Can you hear me now? Is it better?
Yes, yes, it's better.
Okay. So I've changed the internet. I think it's problematic with the word. So I'll go straight to the talk. Share my screen with you once more. Okay. So just let me know if you can see the screen and the
Yes,
presentation.
it's fine.
Okay.
Yes.
So the title of my talk today is actually rewriting the code of life. Um, uh, you know, at some point, I mean, this was a dream for scientists to be able to say that you could actually change the genetic material. Which is the code of life, really, into in a format that you can really influence the phenotypes that an individual is born with. And for a long period of time, it was almost considered impossible that one can really do this. We are a product of our own genes, and how these genes regulate the different properties in cells and tissues is actually how, as an individual, one develops. But thanks to the field of genome editing, this is now a reality. And as I say there are multiple labs who are working in different aspects of trying to tinker around with the genetic code to give rise to phenotype, or to give rise to organisms which are different from what we have come with. So we will understand a little bit about what is the basis of the FELUDA technology before going into more details. So the 2020, of course, he has seen the Nobel Prize in Chemistry being awarded to two fantastic biochemists, Emmanuel Charpentier and Jennifer Doudna, for the development of method for genome editing. It was only a matter of time that the Nobel Prize would have been awarded to CRISPR, because from the day one, it was pretty obvious that it's one of those remarkable things that has happened in the last 20, 30 years, which has really had an impact in every single field that one can think about. And 2020 has been really the year of women leaders in every field, whether it is politics or science. And it's only matching that these two fantastic scientists are actually who have been awarded and been an inspiration to so many different other scientists in this field. It's also maybe a little bit of time to kind of contemplate that a field just doesn't have only two heroes, and that's why I have this additional how to say addition to this slide. Just like Jennifer Doudna and Emmanuel Charpentier, the field of genome editing, particularly with CRISPR, has seen multiple heroes. And somebody on Twitter actually put this just after the Nobel Prize was announced, to remember that several individuals over a long period of time actually contribute to any field, whether it is CRISPR, or whether it could be RNAi, or whatever it is. And several of them have done path-breaking work and are still doing to contribute to this. And of course, not to forget that right on the top you have the picture of a bacteriophage, the virus, anarchy, and bacteria. And they are really the cornerstone of the CRISPR revolution, because we tend to think of microorganisms as less developed, but as it turns out, in some cases they are even more sophisticated than we can imagine, and CRISPR is just an example of that. So what is so special about this kind of system? I just borrowed this slide from one of Jennifer Doudna's presentations. It is a type of immunity, and we are immunity, vaccines, how the body processes different foreign antigens. These are all things that have become very commonplace nowadays. We kind of hear them more and more. But for several years it was assumed that the human cell, or the eukaryote prokaryotic cells are very specialized cells. They are highly advanced, and in terms of the immunity that that they develop. As it turns out, the bacterial cell is not too far behind. And what we see over here is how the process of CRISPR-mediated immunity happens in a bacterial cell, where a bacteriophage, which you see on the top left corner, it's a virus which attacks a bacteria, introduces a genetic material, and this genetic material in the form of DNA is kind of put in a very specific locus of the prokaryotic cell. So you can imagine it's like a librarian, and you have a new book which comes into the library, and you want to put a tag on it so that next time somebody wants to issue that book, you can actually look at the tag, you can look at the acquisition number, and you can issue that book. So it's a pretty similar kind of a principle. Except that it's a very molecularly coordinated event, and you have this piece of bacterial DNA which comes and the viral DNA which is put inside the bacterial locus, which is the CRISPR locus, so that next time the bacteriophage again attacks it, as you see on the right hand side top corner, the bacteria can now get together an arsenal of different proteins and RNAs, which is called the CRISPR complex, which has the Cas9 protein and the guide RNA and so on, and then can go and bind to that bacterial to that viral DNA and chop it off into small pieces. So it's an exquisite immune system that they have developed, and it works very robustly, and this is how they ward off invasions from bacteriophages. So hold on to that thought, because that is going to come back later on, because this is also the basis for how the CRISPR targeting is done. We shift focus to what we call as genome editing. Now, genome editing is essentially composes of three different components. One is that, so here we are talking about changing something in the genome of an organism or a cell. You need to home into that target somehow. You need to add in a modification, and then the new genomic sequence has to be saved in some way so that your DNA is now modified and you have an introduction of some kind of a change. At a very basic level, you can think about this as a Word file or an Microsoft Word file that you're writing, and you're typing a letter, and somewhere in that Word file you have made a mistake. Instead of writing THE, you have written AHE. So if you would have to change that A back to T, you would need a way to take your cursor to that point, to make hit your backspace, delete that A, put in a T, and then save that file. So now if you juxtapose the whole thing into a human cell, or any cell for that matter, genome editing would essentially compose of all these processes. Now, how do you home into a target? How do you make sure that you make a, go into the target, delete a few alphabet, which is the genetic code, and introduce a new set of alpha, nucleotides, and then save this whole information? It seems CRISPR can actually allow you to do all that, and that is because of the highly, amenable properties of this Cas protein. The question rather is actually where you want to do these changes. Are you thinking of doing them in the somatic cells, or are you thinking of doing them in the germ cells, or perhaps maybe even in the embryo? How are these changes being done? Do you think about putting in the CRISPR complex right into the body and doing it, which is the in vivo, or taking maybe cells away from the body and then doing these edits? Or let's say you can think about why one is doing this in the first place, whether we are doing this for disease prevention, whether it is being done for improvement, giving some improved traits, or let's say one is trying to design a few characteristic features. Say, for example, you want to make an individual who doesn't feel any pain. Now, judging from my questions, you can understand that we are slowly going from a point of, let's say, simple biology experiments to very important ethical issues, and hence the field of CRISPR editing is also surrounded by a lot of moral, ethical, and policy making, ethical decisions as well as policy making, and we'll touch upon that later on as well. So this is just to summarize what I told you about. So here you have the bacteriophage, looks like an alien spaceship or an alien of some kind, and you have the DNA which is getting inside the bacterial cell. And once the bacterial DNA comes in, like I said, the bacteriophage DNA comes in, the bacteria quickly assembles this complex of Cas9 and guide RNA ribonucleoprotein complex. Here you see the guide RNA in this two two pieces which gets together, and the Cas9 protein. And this whole complex now goes and sits on the bacteriophage DNA. It tries to base pair with that. And if there is a perfect base pairing or semi-perfect base pairing, then you have this kind of a cut which chops the viral DNA into pieces. So that's the principle, which scientists were smart enough to now take this whole complex and put it inside a human cell. They thought that, if it can do this for a viral nucleotide sequence, what happens if we now specify that guide RNA against a human DNA sequence? And we put this inside a human cell as it goes inside the nuclear pore. And it appears that once you do this, the same complex can perform exactly the same way in a human DNA as well. And you have the base pairing, and then at a precise location after the base pairing, you have the double-stranded DNA break. Now, human cells or eukaryotic cells are actually very, very, advanced in the way that they do not allow the double-stranded breaks to remain. They try to repair it. They put in nucleotides, and there are multiple repair pathways that actually are invoked, and you have the DNA try to, stitch them together. You can do it in a kind of a fail-safe manner, or you can do it in a, something which is very, very precise. And what you see over here is that, most of the times there are mistakes that happen if it is not very precise. But sometimes if you put in a very tiny piece of a foreign DNA, which has got some degree of homology or similarity with the site where the cut has happened, then this foreign DNA can very nicely integrate where the DNA break has happened. This is called homology directed repair. That's powerful because you can essentially introduce any foreign DNA of choice into cells that you want. It could be muscle cells, nerve cells, any other cell at a single cell stage if you put in the CRISPR complex, and you can make a second generation of organisms like mouse and apes and what you can think of into CRISPR edited organisms. So this is the cornerstone. You have the Cas protein, which is the Cas9 in this case, and you have the RNA, which is which we have talked about in which shown in red, and then in black is the human DNA. You have the whole complex base pairing, and then the cut is happening at a very precise point, as shown by the scissors. And like I said, that when the cut happens, the DNA has to get repaired. Most of the time, this is a forceful repaired by putting in the two ends of the chromosome together. At some point, if you put in a donor DNA, then you can specifically integrate a foreign DNA at the target site. That is the part which is really of a lot of interest due to two things. One is that you can actually change DNA the way that you want. You can essentially, this foreign DNA is a matter of your own creation. So you can actually, if you have a mutation, you can put a foreign DNA which does not have the mutation and then bring it back. So you can potentially cure genetic disorders in this manner. And the applications are just whatever field that you can think about, whether it is primate engineering where you put in this complex into sperms or into ovum and you make a next generation of organisms, or you want to target each and every gene that is there in the human body, all the different genes, if and test whether these are druggable, how they respond. COVID drugs have has also employed CRISPR-based screening approaches as well. So all the 20,000, 25,000 genes, the 100,000 long non-coding RNAs, one can do kind of a targeting. This whole complex binds to DNA. So this means that, essentially, if you put in a kind of a signal or a light or a reporter molecule on the Cas protein, you can illuminate the DNA. You can actually see how chromosomes move together, in the dynamic fashion. So if you want to study genome dynamics, this is in the case of TRF2, which is targeting the telomeric repeats, TTIGG repeats. One can actually look at how the chromosomes move about and do these kind of studies. For people who are interested in actually constructing genetic circuits, we have studied about NOT gates, AND gates, OR gates, and how different logic circuits can be built. It appears that you can also kind of exquisitely build them inside living systems using CRISPR. You can make one gene the product of another activation or the product of another repression and build such kind of chemical biology circuits. George Church has done phenomenal work on this field. It's even possible to integrate information into the DNA in the same way. So these kind of genetic circuits. So essentially, you can do a lot of things, and that is why CRISPR is so powerful, that the do's and don'ts of CRISPR as to what you can do and what you should not do is also something that goes along with all discussions about genome editing. And that's because, people have already started taking genome editing into a zone which becomes really questionable, such as that in last year when Jonkai actually talked about making HIV resistant babies. These babies have been born. The question is that in the process of making changes that you have the cell become HIV resistant, what are the chances that you're actually making them prone or amenable to several other diseases like West Nile virus in this case? Or let's say the ethical questions surrounding the fact that the embryo which is getting this CRISPR reagents whose safety efficacy is out of question. These have not been studied. But also, what is the ethical question regarding whether that embryo or when that human being has been born, he or she doesn't have the chance to really revert it. So that kind of a choice has not been given. So a lot of different questions surrounding genome editing in humans and human embryos. So at the moment, there's a moratorium which says that nobody should be doing human embryonic genome editing, or at least even in cases where it is done for certain types of research, not more than the 14-day timeline after which they have to be discarded. So but needless to say, I think I have convinced you enough that CRISPR is something which has actually gone to every possible field that you can imagine. It's beyond biology. It's even into the field of physics and chemistry as well. CRISPR has been tried out in the experiments planned in the International Space Station. One has talked about how entire movies can be encoded into bacteria to think about ways of storing information using CRISPR. There are mosquitoes which have been made sterile and released so that one can control mosquito-borne diseases. It's quite phenomenal how this field has actually shaped up. But perhaps the most important application of CRISPR has been in the case of diseases. And this is still a little bit old. I should have put the latest story from New England Journal of Medicine that came just last month. This is one of the success stories of CRISPR with sickle cell anemia. And a woman, Victoria Gray, was actually given a shot of the CRISPR genome editing complex in an ex vivo manner last year, and the person is doing really well. There are no episodes of sickle cell anemia related complexities within 1 year. Of course, it requires years of follow-up after a clinical trial, but already this is one of those stories which champion the use of genome editing for particularly for life-threatening diseases. And by the fact that the number of mails that we also get from different parents, it seems that this is really the area where one would expect to see a very advanced research in the next few years. Of course, sickle cell anemia is not a disease just of the West. And if you look at how it is spanned across the world, India is far more red than almost most of the countries. More than 50% of carriers are from India, actually. It's caused by a single mutation of glutamic acid to valine, which confers the sickle shaped in the RBCs and increases the hydrophobia, this kind of stacking up of RBCs together, blocking the arterial pathways and thereby having different types of phenotypes. And we started working on this just after I joined CSIR IGIB as a part of something called the Sickle Cell Anemia Mission from CSIR. And our goal in the first phase was, it's now in the second phase actually, but in the first phase, our goal was to see if we can really take this and show as a proof of concept that you can correct the sickle cell anemia mutation. This was before, of course, the clinical trials and everything happened in the West. So to cut a long story short, what we did is that we worked with a different Cas protein, which is different from the ones which almost majority, 99% of the labs are using, which is SP-Cas9, Streptococcus pyogenes Cas9. We used a different Cas protein from the bacteria called Francisella novicida. I won't go into details as to why we chose it. It was a totally serendipitous observation, and we followed up that. But the net effect is that we saw that this particular protein has a very, very interesting property of discriminating off targets by unhooking from these targets, and some of these properties are actually within its structure, and you can make modifications and so on. But the net effect is that it's highly specific. Okay, and we could also take human Indian patients, make iPSCs from there, correct the mutation, and show that really one can do the genome editing in such cells. This story got published last year, and it was highlighted as one of the major stories in that issue. But this is most academic than anything else, because we have a disease here to cure, and we are really, if one has to take this beyond just a lab exercise into a clinical kind of an approach, we would have to do way more than what we saw in that study. For starters, we needed to correct approximately 10 to 30% of cells using CRISPR, and this is a number that several clinicians from across the world have come up to, saying that because the corrected RBCs would have a selective advantages of survival, even if you correct about 10 to 30% of them and not the 100% of cells, one would have a completely disease-free individual. But we were not getting that. We were not getting that same thing when we take a wild-type FN-Cas9 and do the correction. And our plan was that we would take the CD34 positive cells from a patient, do the correction, and return it to the patient. So we really need something which would work to the best in terms of editing. And it was a tall task, because when we thought that how do we approach this, we could if we had to really take this to the clinic, but then at the same point of time in NEJM last year, there was a very interesting paper, an N of 1 trial on a patient customized oligonucleotide therapy, where an individual was diagnosed in 2016, and all the studies for safety, toxicology, functionality, and everything was done within 1 year, which is 2017, and the first dosing was given in 2018. And when we looked at that, we thought that, well, that is a very, very very promising thing to see, because if it means that if all the people who are involved, they get together, and you have the right kind of attitude as far as getting these to people as quickly as possible, then such kind of a trial is also possible. It's just that we have to bring together all these individuals. And we also realized that if you had to take this to trial, we would need, of course, one higher efficiency variant, because the wild type, like I said, was not giving too high edits. You have to solve the major problem, which is that of delivery vectors. We need to find out a way how you would deliver them, because you cannot simply use lipofectamine on patient cells. So that's not possible. And we would also need robust preclinical data to convince the regulators that yes, such kind of a trial can be actually done. So none of this could be done by my lab at IGIV in isolation. So we started collaborating with some of the best people in this business. With Osamu Nureki at University of Tokyo, we started making changes in the Cas protein so that we could make an enhanced or an engineered version of the protein. With Mark Walters and David Martin, we started collaborating on doing some of the mouse studies at UCSF. With Anand Swarup, started developing next generation AAV vectors where we can deliver them. And whom I have not mentioned here is Andre Lieber at Seattle, make adenovectors which would actually be able to deliver this. So I was told that this talk will be streamed on YouTube. So I actually have removed all the work that we have done, but just want to say that we are really happy that we have now a variant or enhanced or engineered versions of FN-Cas9 which work almost 8 to 9 times more efficiently and are currently pursuing the preclinical studies of moving this. There is a little bit of IP involved, so I won't be able to share the details of that. But I'll come straight into FELUDA, because that's where the FELUDA was a byproduct of our work, and all this work and also the work that I'm going to show you is actually done with Dr. Shobik Maiti, is a remarkable scientist. So in IGIV we actually have a kind of a big lab together. He brings in his biophysics knowledge, I bring in the molecular biology knowledge, and we try to tackle some of these questions together. FELUDA was actually a diagnostics that we developed using FN-Cas9, understanding that it's a very specific enzyme, so you can use it for distinguishing nucleotide sequences by one mismatch. Why the name FELUDA? Because there were similar CRISPR diagnostics that were already available from MIT and Berkeley. They called it Sherlock, they called it Detector. So we also thought that we would have a very Indian name, and when you have 2 Bengalis in the team, that's the byproduct of that. And we the full form is FN-Cas9 editor-linked uniform detection assay. And it works on the principle that, like I said, this Cas protein is highly specific. If you indirect it to a specific nucleotide sequence, it can differentiate between two nucleotide sequences that are different by a single mismatch. And the readout can be on a gel, it can be on a capillary electrophoresis, it could be a fluorescence-based or a lateral flow device, and so on. And then I will talk to you a little bit about the lateral flow later on. And you can distinguish between two nucleotide sequences. The way we arrived at this was that we could synthetically put mismatches on the guide RNA sequence of the Cas protein, which I showed earlier, to give a completely different outcome between two targets. In this case, the wild type and the sickle cell anemia substrates are shown, which are different just at the point where the A to T mismatch has happened. And as you can see, the wild type substrate is not cleaved, whereas the sickle cell substrate is completely cleaved or near completely cleaved. So this we converted into a readout, which would be robust enough to work for every single time, and also to actually detect heterozygosity as well. So we did blinded assays on patient samples for sickle cell anemia carriers as well. And currently, carrier screening can only be done using sequencing, which normally take around 20,000 rupees or even more, and several days to finish. But FELUDA showed 100% concordance with the sequencing results. One can actually convert this into an affinity-based readout as well, and also for a PAM independent version. People who are in the CRISPR field know that Cas requires a particular sequence called a protospacer adjacent motif. So we converted that into a PAM independent fashion. While we were working with Dr. Govind Makharia at AIMS, so he came and told us that there are patients who come to his OPD who have Helicobacter pylori infection, and this causes the peptic ulcers. And normally what they do is that this particular bacteria, depending upon a variant strain that this bacteria sometimes has, are either rifampicin positive or rifampicin negative. It's a first-line antibiotic. And what they do is that they culture this bacteria for 3 days on plates, and then they'd say that, okay, you have got a rifampicin resistant version because you have that variant, and then they give the corresponding antibiotics. If one does a FELUDA, then this can be done within half an hour or 45 minutes while the patient is sitting at the clinic. And we put that to test on the field just before the COVID pandemic had started. The team here, you can see Dr. Shobik with an individual in a tertiary healthcare center, where the government really does these kind of tests called solubility tests, where the coagulation of the blood happens and gives some idea whether these samples would be then later on go for a pulse field gel electrophoresis or a sequencing or so. It's a first line test for sickle cell anemia carriers and individuals. Very, very crude, not at all accurate, and it requires blood to be pricked and drawn. In this case, you can see these kids here from a primary school. And one of them, this young girl over here who doesn't have shoes, is not too thrilled at the prospect of having the finger pricked. So we came up with an idea where molecular biology 101, just this have been done before, where you have certain chemicals in the tube, and the child is just asked to spit into that tube. The cells break, the DNA comes out, and the FELUDA can be done immediately, and the results can be obtained whether that mutation is present on them. So this was where we were at the beginning of the pandemic. And of course, then the COVID-19 struck, and we figured out that if FELUDA can work for point mismatches, it should work even well if there are 20 bases of match, which would be what we needed to detect in the SARS-CoV-2 genome. So essentially, this is the pipeline for FELUDA for SARS-CoV-2, where you have the first part, which is similar to that of a quantitative real-time PCR, except that we do a RT-PCR here, which is a reverse transcription PCR, single step, and then using a normal PCR machine, we do an amplification reaction using biotin labeled probes. And these biotin labeled probes generate a biotin labeled amplicon, and then we combine that with a paper strip chemistry to give rise to a result which can be obtained within 45 minutes to 50 minutes. Of course, it doesn't require a quantitative real-time PCR machine, so testing can be scaled up 20 times. That's the cost difference between a normal PCR and a QRT PCR machine. And it also has semi-quantitative flavor, and it's relatively rapid. And it's very specific because I told you that the FNCAS9 protein is highly specific to its target, and it can closely differentiate between targets such as SARS-CoV-1 or MERS or influenza and so on. So won't go into details of the chemistry. There are gold nanoparticles that move, and there are biotin labels, tryptophan biotin interaction. But the net result is that you have a readout like this. If you have a positive sample, which would have 2 bands, if there is a negative control or a negative sample, then this test band would be invisible. So it works similar to that of a pregnancy kit, except that in a pregnancy kit you have an antigen antibody interaction. In this case, it's a DNA protein interaction, highly specific one, which it can distinguish. And when there is a FELUDA, there is, of course, it's a sacrilege if you do not have Topshe and Jotayu. So we teamed up with a startup in Chennai to come up with a smartphone app where one can simply take a picture of this strip, and this is machine language learned, trained, and then it can give a readout whether it's a positive or negative. So you don't really have to depend upon your visual interpretation. It's very sensitive, goes down to just a few copies of the virus. And to help people generate data for other diseases, we have now Jotayu, which is a junction for the analysis of target design for your FELUDA assay. So Jotayu is basically a web server that allows you to predict and design guides and the entire FELUDA experiment for any disease of choice. And of course, FELUDA doesn't require a PCR machine in the newer versions. It can be done using isothermal PCR, where you do not need the PCR machine. You just need a heating block or some kind of a water bath maybe, and all can be done in a one-pot reaction. This is a slightly older cost table of the reagents. It costs just about 300-400 in terms of reagent costs, and the time taken is also quite low. This is just a small part which is just to give you a little bit of a flavor about what it means to go from such kind of an idea to a product. Some of the lessons learned. Of course, FELUDA has got a lot of traction. I believe more because of the name. The recent email came from Anandu Publishers, so you can imagine where this has gone to. But it was very interesting to see. It's a bit. I would say it's a bit of a distraction, because there is similar work that is happening from all over the country. Lot of amazing scientific things happening, more related to COVID. So at some point it was a little bit of a distraction, but yeah, it was very interesting to see how what are the challenges in actually moving from getting an idea to a product. And that's what I'm going to tell you in the next few slides. I'm almost done. So of course, ICMR approved this in September, and interestingly, they said that if you do a FELUDA test, or in this case the Tata MD check, which is the official name of the kit, then you do not need to do a qPCR test. So it is at par with a quantitative PCR data, and it's also one of the first, if not the only one at the moment, paper strip based CRISPR based diagnostics. So what are the things that we learned during this whole process? First of all, this whole thing was actually squished within a few months, and that's really the timeline that we were working. 20th January we had the idea, and September the DCJ approval after a new company was formed, and the patent and licensing was all done within that period of time. Just to tell you, this is indeed true. This is a screenshot from my WhatsApp chat, where we formed a group on 28th January, where even before the first strain was reported in India, and sometime around March, we had one of the first samples in Delhi that came at NCDC for COVID positive sample in India, where we could test this prototype. It was a kind of a eureka moment. We have the control band developing there, and then at the bottom you will see now very soon the positive band developing. So this was one of the first samples that came to Delhi, and the RNA was extracted. And then of course it was approved in sometime in September. Today in the afternoon, just before I came in, it gave me a kind of an eerie feeling, because I just got a call to collect 2 samples of the UK variant, which has come to IJIB. And just after the talk, I would be rushing to the other campus to get this, because we have what I showed you in some of the slides. The real USP of FELUDA is not just detecting nucleotide sequences, but actually distinguishing between point mismatches. So as you can imagine, that the UK variant is all SNVs, right? These are variants of one mismatches. So what normal RT-PCR might fail to catch. That's why they are doing sequencing of these UK variants to understand if they are carrying that mutation. We believe FELUDA can actually do that very efficiently, and that's why we are developing FELUDA at the moment for the variant. But one of the things that we learned during the entire process is that we speak a very different language. Scientists speak a very different language than the language that companies speak. Most of them they want risk-based technology because that gives them an edge in time and in budget. And we are almost most of the times we do not know how to address that. Thankfully, in this case, we need good translators, and our director, Dr. Anurag Agrawal, has been in the news on multiple policy making decisions. Had been a real boon to have him on the team. So he was like the Google Translate. And we have done close to 4,000 5,000 samples with FELUDA. We understood a lot of different things in the process. Number one, we need to have reagents that are good if you want to have something which is very robust. And sourcing these reagents was a challenge during the pandemic, that you have the exact same thing in terms of quality. Most of us are molecular biologists. We know that how differences in quality of PCR reagents can actually make changes. The quality of the RNA that is extracted, even that has an impact on the quantitative real-time PCR. And most importantly, the quantitative real-time PCR itself being called as a gold standard. And I won't go into details as to how many kits that we have tested now, and we realize that QRT PCR is so much dependent upon the kit that you are using. And sometimes they are not even bronze standard to actually understand whether we have a 100% concordance. So the good thing is that we had the possibility of doing sequencing. I think IJIB has contributed probably more than 50% to the Indian sequencing efforts of COVID-19 genomes and the patient genomes. So we had in every case we could go back to the sequencing data and also see how much of how much of sensitivity we were getting. The other thing we learned, and this is really important, I make sure that I put this slide in every talk, is that we underestimate the power that our students have. And these are students who actually stayed back during the entire pandemic. Ever since the COVID-19 broke out, the lab was not closed for a single day. Several of them actually did not go home. They stayed in the hostel, contributed to the work, and they are even doing it now because of I just talked to one of my colleagues who has the bed, mattress, everything in the office because they are sequencing this UK variant. So, and in the end, it's the students who do this work. It's a pity that, very, very small things such as releasing fellowships in time and actually taking care of their, some of these very basic things are sometimes overlooked. I just hope that this pandemic, puts science in a completely new light before our administrators and policymakers. If we can retain some of these very bright students, then there is no reason why we should not be performing the same way that some of the other labs across the world are performing. So yes, our students are our biggest asset. The third thing, of course, is we have heard that raising a child requires a village. In our case, we learned that if you have to bring a product from an idea to a market, it requires coordination and cooperation at so many different levels. And it's right from the courier person who handles the Merck oligos during the pandemic to get it cleared at customs, move into IJIB, to the office of the director general or the principal scientific advisor. Everybody who was a part of this, so to really make things smooth. And I'm really so fortunate that so many of people came together. The entire CSIR came together to try and help us in bringing this out. FELUDA got a lot of attention, some of them not very good. It was a very interesting experience because I never understood that science would have so much attention. There were a lot of trolls on Twitter and Facebook and abuses and so on, because of course I think perhaps because of the media attention that it got. but also very, very, hearteningly, I think the entire pandemic has put science much more accessible to make science more accessible to public. It was very, very heartening to see that FELUDA at some point in September was trending just after Sri Krishna and press freedom in our country. That's really, what more can we expect from if you're as a scientist? And when Shashi Tharoor also, quotes it without using a single bombastic word, it felt really good that, if you have a discussion, a sensible discussion now in the streets, I think people know about vaccines, people know about immunity, people know about RT-PCR. So if anything that has moved ahead and has been championed during the pandemic, it has been science. So it's quite wonderful to see what that can do. And finally, pandemic gives a kick in the back, so it's a mother of invention, and the lab has been working on all these different ways how to make it possible to do FELUDA closer to home. Currently, of course, Tata Medical and Diagnostics are doing this through their kits, and it has to be still done in a lab. But the idea was that, can we make it come closer to the home, in the office, or maybe allows return to schools and colleges? And actually, the students came up with ideas that you can do this kind of in an equipment-free manner, where you basically have all these things pipetted you would not require these pipettes. Actually, they would all be pre-pipetted into small tubes. But what I essentially want to say is that once the CRISPR components are added, you can simply flick these tubes. You'll see here the sample is going in. You can do this using even a dropper. You don't need a pipette. You can simply flick these tubes to mix them. No need of a centrifuge. And you put it inside the elbow, and you are using the body temperature to do isothermal PCR. And then you place the lateral flow buffer, and followed by the strips. And then you have your readout within 25 minutes or 30 minutes, whether one is a positive sample or not, or a negative sample or not. So that's where we want to go to in the next few months. We have some ideas about how that can be implemented. All depends on whether India is ready to have such kind of simpler tests which can be done at home. That's a different complexity altogether. But that actually is my last slide. So it's a large group that has worked, and it's a large group that is working on it. Like I said, we are busy with the UK variant right now, and a lot of collaborators, a lot of people who have helped. And we are also very generously funded. Tata MD is, of course, the one who has taken this the most as close as possible for commercial applications. So if you have any questions, this is a good time and to ask me and let me know.
So are there any questions?
Hello.
Yes, go ahead.
Hello. Thank you so much, sir, for this discussion, and it has been a great from hearing the actual inventor of FELUDA. So my question is actually my question is that I would like to know actually which protein did you target of SARS-CoV-2 for getting the match in FELUDA? And another question is that if there is a mutation or big strain variation, will that FELUDA work?
Yeah, so we are targeting the S gene. Let's in the Tata kit, they are targeting the S gene, and this is an ongoing process that we have an update every single week. We go through all the genomes that are being deposited. Now it is over 100,000 genomes of the SARS-CoV virus to make sure that there is no mutation over there. We have a backup of different other probe sets. The same principle applies to any qPCR kit essentially. So it has to be continuously updated. Fortunately, we have taken a region which is not prone to mutations and currently doesn't have any mutations. But yeah, it's a process that we are continuously doing as a quality check.
Okay. Thank you, sir.
Hi, Devajyoti. This is Kaushik from Bose Institute.
Hi.
Great talk, great invention, great product.
Thank you.
Just one single question out of curiosity. I have come across certain publications which have shown that similar strip-based assays using CRISPR-Cas system were done on Cas12, Cas13 proteins, which I think they have a promiscuous DNA cleaving activity, and they utilize that promiscuous activity of those Cas systems to do the strip-based detection. For the FN Cas9 for your FELUDA, is there any promiscuous activity that was utilized?
No. So the Cas12 and the Cas13 detection systems, which are called the Sherlock and detector systems from MIT and Berkeley, they actually rely on the trans-cleavage activity of reporter molecules. So essentially, once they get activated upon finding their target, they start cleaving DNA or RNA reporter molecules which are in the vicinity, and that gives a fluorescent signal or some kind of a chemiluminescent signal. FELUDA actually works on the principle of direct binding. So there is no reporter cleavage involved. Here we are looking of direct stoichiometric binding of a protein to its DNA target. So of course it meant that we needed to optimize things at a much more stoichiometric level, because here we are talking about single DNA molecule with a single protein molecule binding. But the net result is that what you are seeing is a semi-quantitative readout. So if you have 100 molecules of Cas protein and 100 molecules of DNA, your signal intensity would be directly related to that. In the case of cleavage based thing, a single Cas12 or a Cas13, if it gets activated, will cleave all the reporter molecules in the vicinity. So you would not get any idea about the CT value or the viral load in that sample. All of them would be pretty high in terms of the fluorescence. So that is the fundamental difference between the two formats of detection.
Okay, got it. Thanks.
So any more questions? So if not, let's thank.
Yes, ma'am, I have a question.
Okay, but go ahead. Yeah.
Yes, thank you, sir. This is a very informative and very nice work. I have a very general question.
Introduce yourself.
Myself, Sharmishta Majumdar.
Hi.
So, my question is from the side of therapeutic level. So, as you shown in your slide that sickle cell anemia treatment and Casper based treatment. Here, I am curious to know that in case of in vitro, yes, in vitro state, I understand that if you are putting the Casper treatment in the petri dish, so it can work. But what I don't understand, that is the in vivo state. Means how if a patient can be recovered. Means we have cells, means so many cells. So if you are targeting a specific cell or how it work, means it will go to the bone marrow cell to change the mutations or like that. Have you got my question?
Yes. So that's a very valid question. How would you make sure that all cells of the body have the mutation, right? That's the problem. Fortunately, you will have the first diseases that will be cured with CRISPR or with gene therapy would be the ones which are blood cells. And the reason is exactly what you said, that you need to find out a niche cells or a group of cells that, if corrected or changed, would lead to all the cells of the blood being corrected. Okay, and these cells are the hematopoietic stem cells, which happen to be in the bone marrow. So the treatment using gene therapy or gene editing is actually done after something called myoablation of the bone marrow, where all the niche CD34 positive cells are removed, and then your edited cells are engrafted in the bone marrow. So what happens is that once they get engrafted, if they are successfully engrafted, they keep on producing the erythroid cells and the progenitor cells and the blood cells after that. So So therefore, it's a one-time injection which leads to all the blood cells being corrected, and the net and also the resultant blood cells will be of two types. One, of course, is your sickled, which is originally there inside your body, and your RBCs, which will be corrected. Now, how do you take care of the fact that there will be also sickle cells in the patient? Is because your RBC lifetime for a normal RBC is about 120 days, whereas a sickled RBC stays way smaller than that. It's about 30-40 days or so. So because of this selective advantage that your corrected cells will have, they will slowly start taking over all your blood cells, and thereby the patient will get cured. Of course, it doesn't happen the same way with other diseases, such as muscular dystrophies and all, where people are still trying to find out ways by which you can deliver them specifically into muscle stem cells or things like that. But for blood, it is pretty much now a procedure which can actually be followed.
Okay, that means it will take some time to get recovered from this.
So like I said,
Means
particularly this patient that I talked about, New England Journal of Medicine article, this is 1 year and she has not shown any symptoms of the disease anymore. So it's a remarkable recovery.
Okay. Okay, now got it. Thank you, sir.
You're welcome.
So any more questions? So if there are no more questions, thank you, Debojyoti, for this extremely illuminating talk in a extremely short space of time. I think now you would like to prepare yourself for the next exciting part of.
I don't know if you are still interested. This is something that.
No, I think. Yes, we are. Of course, we are. So we give him some time.
We need to give some time to Debojyoti.
Yeah, maybe 5 minutes.
So if you have any comments, we'd be privileged to listen.
Okay, so let's give Debojyoti some time, and let me see if I can get a small presentation up here. So I thought I would introduce the other two techniques that we have, CRISPR-based techniques for detection. Baba, manage korbo. Exit korbo ki kore এখান থেকে?
প্রেজেন্ট নাও করুন, ওই প্রেজেন্ট নাও থেকে তারপরে স্ক্রিন সিলেক্ট।
প্রেজেন্ট নাওতে ছিল তো।
কার্সারটা সরালে নিচে প্রেজেন্ট নাও অপশনটা আসবে।
না, সেটা জানি আমি। Share your entire screen তো হলো, then এখানেই তো হবে প্রেজেন্ট নাও। Present now.
Yes.
What is appearing? Nothing is appearing. So I can't see anything.
Now we can see the slides. Yeah.
What?
Presentation mode. You can go to the presentation slide show.
What F2?
अभी आप प्रेजेंटेशन ओपन कीजिए, है ना? Stop करके प्रेजेंटेशन.
Doesn't matter. Anyway, I think even I think if you can hear me, it will be fine. So I was talking about the two other methods that we have for CRISPR-based detection of SARS-CoV-2. which already Debojyoti has alluded to. So as Kaushik was referring to, a lot of these diagnostic methods have been So I think even if you can't hear me, it'll be fine if I just explain the two other methods. Can you hear me?
Yes, we can hear you.
Yes, we can.
You can. So I can just introduce the two other methods. So these are based on the promiscuous collateral activity of Cas13 and Cas12. So Sherlock is one of them, where it is detector was the technique which was developed at Berkeley, where you have this Cas12a protein, which basically exhibits non-specific collateral activity and degrades all the DNA molecules that are adjacent to the target RNA. So what happens is you mix your Cas12a protein targeting CRISPR RNA with complementary single-stranded DNA reporters, which are fluorescently tagged. And once you have cRNA-dependent recognition of the pathogenic nucleic acids by Cas12a, then the collateral activity is turned on and there is destruction of the DNA probes. And the moment that happens, these DNA probes are similar to conventional Tachman probes. So one end of the reporter is bound to a fluorophore and you have the opposite end linked to a quencher. So degradation of the DNA probes actually releases the fluorophores and you get a fluorescent readout. And similarly, you have Sherlock, which is based on another CRISPR-Cas system, which is Cas13, where you have collateral activity is basically an RNase activity. So this is how the two systems, Sherlock and detector, works. And basically with Sherlock, they have detected Zika virus, dengue virus, pathogenic bacteria, and single nucleotide polymorphisms in DNA with attomolar sensitivity, actually. So this is just to give you a glimpse of the kind of progress that is being made in terms of diagnostics. And of course, sensitivity is the primary advantage of these techniques. So Sherlock is capable of detection down to 2 attomolar of both DNA and RNA targets. And of course, as Debojyoti has already been telling you, it's the simplicity of these assays where you don't need PCR machines. You can perform these assays in 5 to 10 minutes. All you need is an isothermal amplification step before you go to the detection step. And this detector is being sold now already by Mammoth Biosciences. And Sherlock also has been developed into these paper strip lateral flow readout assays for Zika and dengue virus. So this is what I can add to what Debojyoti has been talking about, that these diagnostics are being developed with a lot of rapidity, because of course diagnostics are free of the ethical issues that are associated with other forms of genome editing, as you've already heard. And of course, other than that, Debojyoti has pointed out some very important issues regarding how science is run in the country and how that will either impede our progress or accelerate it. And I think these are very important points to ponder about also, because I think it's only if groups in general can build on collaborations, can work together, can facilitate its science at all costs, in spite of whatever bureaucracy is there, that we can actually move forward. And that's, I think, a very important lesson to be taken, particularly here in the surroundings of this particular city, where we're really bogged down in a lot of paperwork and bureaucracy, and it's very, very difficult to make progress at times. So I think if anyone else would like to add to the discussion, you're most welcome.
Thank you, Professor Basu. And we'll have a special event now at the end of this webinar. And I guess Debojyoti said he is ready. We have reached almost the end of this year, 2020. We had one extraordinary year almost throughout this year, we had this pandemic and we are locked down at home. And two things mostly have helped us. First, definitely science. And the second is music, literature, and various other forms of art collectively. So to me, science and art, they are inseparable. So if we celebrate with science, then why don't we celebrate also with art, music, and anything else that we could? And we are fortunate to have Dr. Debojyoti Chakraborty as a speaker. You'd be happy to know that he's also an eminent musician in the Indian classical music field. So he has kindly agreed to give a short demonstration on his musical endeavors, activities. So Dr. Chakraborty, over to you.
So thank you, Shubhroda and Dr. Ghosh. I think I've never done this before, so it's it's an experiment for me. I don't know how this will pan out, but I think there was a request. Dr. Dostidar himself is a very very good musician. I have heard some of his renditions, so I'm also a bit scared what I'm going to play because he will be being there in the audience. So I don't know what is the best way to do this, but anyway, I'll try. Like he said, that science and music are indeed inseparable. They are complementary to each other. And what I have with me today is an instrument, which is an Indian instrument. And several of you have heard. For the people who have not heard this instrument, it's the sitar is something that that has been used in different formats, not just for classical music, but also for semi-classical and other things. And there are reasons why this instrument is used because of the range and the diversity of notes that you can play. And it will be a very short short session. I can just tell you a few things about the instrument. So like I said, that Indian music or Indian classical music is predominantly based on vocal music. So that's how most of our music actually originated. And then it was important that there would be certain instruments that can give, as far as possible, the same elements of vocal music to make the exposition even better. So what do I mean when I say that it is that the elements of vocal music? So the difference between the Indian music and several of our Western music pieces is the fact that their instruments are a bit limited. Several of their instruments are a bit limited in scope and application. So if you would have a piece like let's say so this is a very famous symphony from Mozart, and these notations are generally written for the piano or the organ, and that's how the Western music developed from the church music actually was written down. And there is no way to go beyond what has been noted down. So if you ever hear a Western orchestra, this is what you probably get to hear. But the fact that in the Indian music there is so much of prevalence for semitones and glides that you would interpret it in a very different format. And from that comes the fact that what is it so different in what I played right now from the staccato type music. So it is not just the fact that there are glides and there are these semitones, but also the emotions that it it kind of brings inside the listener. So as a PhD student, most of you are PhD students, I think, from what I see in the names. PhD students' life is a very, very heterogeneous kind of ocean of experiences. And some days when you come into the office, you have good times, and some days when you come, you do not have very good times. So your boss might be right there sitting, in the room, and telling you that, well, I need this experiment to be done immediately because I have to send a presentation. So, the boss might have a very different way of talking, and it could be. And she expresses her emotions, he expresses her emotions, but this expression of the student is not necessarily the same. And you might be, if you're a first year student, you might have a. Because you do not know what to say, right? You have been given something to do, and you said, "Why me?" Can you give it to the other project assistant, maybe? Because I have so much to finish. But that doesn't happen. And then suddenly, you have your senior coming because he has now got a new postdoc offer and he's all bubbling with excitement and happiness, and he simply goes and says that. And the mood is light, and he said, "Oh, chalo, let's go." I don't know if what was his name? Was it Zakirda who had his tea stall next to the campus?
Yes,
Zakirda. There or not? And he makes this amazing muri and with omelette, and they go for some tea and there and have some sessions over there, and you are discussing everything starting from Marx to Lenin to possibilities of a ray thrown hereby, and somebody might come and say, did you hear that music in Pothe Pachale? I think what I heard sitar today, it is something similar, and then somebody just talks about that. And somebody comes with a different idea who is a very, very, let's say, aloof from all the metaphysics and all the things, and says that everything is because if you are having a religious bent of mind, then that's how things are becoming correct for you. And he is somebody who listens to bhajans and devotional things and comes up with his own format. It's you see, different types of people have their own moods. They bring in their own, philosophies into a lab, just like they bring into an institute and a country in different ways. But that's how diversity develops, and there would always be that one person who is very, very sophisticated. Among Bengalis, this is very common to have somebody who would be the one who would be only listening to Western classical. He doesn't have so much of idea about Eastern music. He says that no, these are all crap. You should listen to the very cultured Western music pieces. And he says that have you heard about let's say Furellis from Beethoven? And in his mind he's always playing Furellis. And then there would be that Indian guy who would say that, what type of music you are listening to? It is so boring. Let us make it a little bit more funny. And he gives his own ideas, and with his own Kirwani and Yaman Kallan and whatnot, he brings into the picture. And then in my lab particularly, now that we are doing a lot of these FELUDA experiments, sometimes you would have this bad batch of millennial strips coming, and you would spend most of the time understanding what is happening. Is it the assay is not working, protein is not working, and then you would have a little bit of let's say Sham Kallan representing the mood better, because you're confused, right? You do not know what is that experimental phenomena that you need to correct to get it work. Till the Shuddhagandhar and Madhyam come in and tell, oh, okay, this was a bad batch of protein. So you have to catch that hold of that guy who made this protein. That moment of realization, And then somebody says, but we used the same batch last time also. I said, no, but wait, it could be the same problem. So this is Indian music, it is always, full of aesthetics, full of realizations as a musician, as a listener. And when, all the things work out completely and you have the right FELUDA experiment coming in, then you can, go to the nearest restaurant. Now, of course, you cannot go to the restaurant, you have to order in, and then in your mind, you have something else playing. So I think that's just to give you an example of a typical scientist's day, a typical student's day. And it's no way different from a musician's day, because musicians are out of jobs. They are all freelancers. So I think they are finding their best possible ragas right now to, talk about their movies. But yeah, that's.
Lots of applauses for Devojyoti.
Thank you
I
very
saw.
Yes, I found entire my PhD and postdoc experience in your sitar strings and
Oh, that's nice.
vibrations.
It's not very different from scientist experience. You ask anybody here.
Exactly. So very nice, very nice.
Okay, so
Many thanks. I mean, no thanks would be sufficient. Devojyoti, would you like to make some comments?
No, I'm spellbound really. So I'm at a loss for words, but really it's been a wonderful afternoon, and thank you so much for sharing your experience with FELUDA, with your music. It's been a great pleasure having you with us.
Thank you, ma'am. It's been
Taniyadi.
Yeah, really from the very beginning, first of all, almost like Satyajit Ray, you guys have, projected FELUDA and Jatayu and Topse, all of them. Excellent work and great talk, Devojyoti. And then because I always feel that if someone is creative, you have creative brain, not only doing science, music and science are actually should go side by side, about which myself and Shubhro, we almost every day discuss, and always we find solutions from the music part. Being a vocalist of our country, I always try to find out that at different at any or every of your mood and situation, you will get some Tagore's song. Always I get that, and from your this music, I now thought that okay, I will arrange also in this way, maybe for one of my performances in some of the stages. And actually, I never thought that the way you explained all this, gave explanation. Really, hats off to you. I'm really proud.
So,
Taniyadi,
I must say that this idea
I
is
think you
also
have
a bit stolen.
the idea.
No, this idea is also a bit stolen. I think I've been very much influenced by Ustad Zakir Hussain. He does his he tries to bring in all these elements also in his performances. So it's a bit learned from him also a little bit. Nowadays it's very difficult to catch hold of him in India, but whenever we get a chance and he's giving lecture demonstration, it's something that we learn from the greats. But now I'm a bit scared because there are so many nice musicians and vocalists there. I don't know what how you would be.
Excellent.
Monikul bhai কিছু বলবেন না?
I'm totally spellbound. I was aware that he is a sitarist, good sitarist, but I have never heard. So it's really, really enjoyable. And I have gone through your papers when the FELUDA came in the market. So thank you very much, Devojyoti. Try to visit Bush Institute when all this pandemic will be over. Thank you.
Professor Lahiri Majumdar.
Well, I heard Devojyoti before. He gave a talk a few weeks ago, under NIBMG at BIMT probably, very similar to the talk that he gave today. But definitely it's much more than what he told there. But I'm absolutely certain, but I'm flabbergasted by the way you presented your sitar. Shubhro, in fact, told me yesterday or day before yesterday that you are also a sitarist. So I know Shubhro for his cultural part as well. So I was extremely interested to see how it goes along with your science, which we know a little bit. And what Tanya said about science and creativity and science and music, I'm absolutely in favor of that. Because I myself follow that, and I'm very much interested in creating a situation in which research, which is science, is nothing but a creative phenomenon altogether. And this thing cannot go without the creativity of the music. They go so well And I'm extremely happy to see that one of the young guys at IGIB, I don't know, so for me, at one time, for me, Brahmachari was at one time, he was very interested in music. He couldn't sing, but he couldn't play anything, but he was interested in that. I knew that. And I don't know about Anurag, how he feels, but definitely he's a very good scientist.
I think he's very far away from music.
Right. Shomir was different. Shomir Brahmachari, your founder director, was very different in that case. Anyway, this is extremely well. And Shubhro, I congratulate you along with Debojyoti for organizing his talk here and along with this idea.
Jhumur took this lead.
Jhumur took this lead. Well, I know the group, young group of both institute. Very happy to see that they are really doing very, very well. And creative group here. And extremely happy to see these things together. But Debojyoti, please continue with your music as well.
Thank you, sir.
Feluda made you famous. But I think no worse than that. Please. The way you explained, I like that.
Yeah.
I only had my music class this morning. I was full of music, and I was rushing to attend this lecture and your music.
Thank you.
Over to talk to Sudipto Shah.
Yeah. So at the end of the talk webinar series, 5th webinar series, and on the behalf of the Division of Bioinformatics, Bose Institute, I thank today's speaker, Dr. Debojyoti Chakraborty, for a wonderful talk and the sitar music. I would also like to thank our Jayati Di, Professor Jayati Basu, for chairing the session, the 5th session. And I thank all the participants for attending the 5th webinar series. Hope you all enjoyed the talk as well as the music. So our next webinar speaker will be Dr. Nimesh Gupta from National Institute of Immunology. He will talk about vaccine, specifically focus on the vaccine against COVID-19. So till then, stay safe and goodbye, and wish you all a happy New Year 2021.
Happy New Year to all of you. Happy New Year.
Jhumur, before you conclude, let me tell you one thing. Shubhro, you can take initiative in getting Monikuntala, who is a very good Nojju speaker. Apart from Anya and others, you do not know that Monikuntala is a very good Nojju singer.
I came to know very recently. I knew about her interest in music.
Shampa Das of DPB is also a sitarist.
Oh,
you probably didn't know that.
No, I didn't know.
You didn't know that. Shampa is a sitarist. She plays along with Sugata Marjit.
Oh,
okay.
Okay, so please include these in your future seminars. That will help. Okay, good luck, everybody.
Thank you. Thank you. Bye bye. Yeah.
Bye bye. Happy New Year. Yeah.
Bye bye. Happy New Year.
Happy New Year.
Bye bye.
Wow.